Okay, well, welcome everyone. It's a pleasure for me to introduce our speaker, Dr. Seth Newsome. But before I do, I just want to quickly run through some of the basic ground rules. So all audience participants will be muted throughout the seminar. Please do not attempt to turn on your video or share your screen during the talk. There will be an opportunity for the audience to ask questions live at the end of the talk, just like a normal seminar. Once the speaker is finished through their talk, you can use the Zoom raised hand feature to modify or to notify the meeting host that you'd like to ask the speaker a question. The raise hand feature is located under the reactions tab at the bottom of the Zoom window. I would also remind you that if you send a message to in the chat window, that it will only go to myself and Emily. So we can relay the question if you have one, but it's easier if you use the raise hand feature, and then we can unmute you. And also, we would also ask that you show your face by undoing your video when you ask your question. It's always good to put a face with the name. Okay, so it's a pleasure for me to introduce our speaker today, Dr. Seth Newsome. He is an associate professor at the University of New Mexico. And you might be wondering, why would somebody in Mexico be doing a talk on marine systems? Well, Dr. Newsome has worked on a variety of systems throughout his career. He's currently the associate director of the New Mexico Center for Stable Isotopes, and he's an associate professor in the Department of Biology. He received his undergraduate degree at Dartmouth College, and then he went to UC Santa Cruz and did his PhD with Paul Koch, and working on looking at ancient pinnipeds. And then he's also conducted a number of postdoctoral appointments at a variety of different places. I've always known him kind of as being the stable isotope person, but he's quite varied in what he does, and I'm anxious to hear what he has to say today. So today he's going to talk to us about a multidisciplinary approach for assessing the vulnerability of species to environmental change. Welcome, Dr. Newsome.
All right, can you guys hear me? All right, thanks, Scott, for that introduction and the invitation to give the seminar today to Moss Landing. I've worked with a couple of folks there. Scott Hamilton, I think I saw him on the list. Hi, Scott, and a Mike Graham as well. But anyway, I'll go ahead and get started here. So to provide some conceptual context for my talk, I will use a recent review by Dawson et al. That argued a species' vulnerability to environmental change is the sum of three primary factors. The first is exposure, the second is sensitivity, and the third is adaptive capacity. Now, while this paper focused on individual species, it's important to remember that this framework could easily be applied to whole communities or even ecosystems. If you plot these three factors on a single graph, a species' sensitivity is essentially the inverse of its adaptive capacity. Vulnerability increases with a species' exposure and sensitivity to environmental change. And what really determines how sensitive a species is to change, or how much adaptive capacity it might have, is related to a combination of traits, including life history, genetic diversity, phenotypic plasticity, dispersal colonization rates, and resource and habitat requirements. Now, I've highlighted the three in red because they're kind of the focus of my research program. Now, another important aspect of this equation is the fact that we are currently living in an ecological bottleneck for both unnatural and natural reasons. For example, what we've learned about the ecological plasticity of marine mammals over the last century or so is likely a subset of their possible behavior for two reasons. The first is that most of the extant species in this group evolved and thrived during the Pleistocene, which is a period of Earth's history that's known for its environmental variation. And the second reason is that the unnatural degree of historical human exploitation experienced by marine mammals, which were hunted and sold as commodities for centuries prior to their protection in the late 20th century. This persecution reduced their population sizes to a fraction of what they were just a few centuries ago. So going forward, as species experiences a larger array of environmental pressures, it would be good to know what species are capable of in terms of their ecological and physiological plasticity, which by extension defines their adaptive capacity and may enable us to better manage them during times of rapid environmental change. Now, I argue to do this, we need information on three distinct but related realms. First is work on historical ecology that examines ecological and physiological plasticity over evolutionary relevant timescales from centuries to millennia. Now, an example of this is my work, dissertation work actually, that reconstructed the ancient biogeography and life history of northern fur seals. And by using a combination of morphometric and isotopic information, found strong evidence that this species had extensive breeding colonies at temperate latitudes not so long ago, from Southern California all the way up to British Columbia. We also found that fur seals breeding at temperate latitudes in the past used a longer lactation strategy than their modern counterparts that breed at high latitudes in the Bering Sea. This long-term maternal strategy is very similar to that used by California and stellar sea lions that dominate the odorized community at temperate latitudes today. Another more recent example is work on North Pacific albatross, specifically short-tailed albatross. We used a combination of bulk tissue and amino acid isotope analysis to identify the foraging grounds used by modern and ancient short-tailed albatross populations. And not surprisingly, found that ancient populations spent a lot more time in the California current than their modern counterparts. We also found that modern short-tailed albatross appear to occupy a slightly higher trophic level than their ancient counterparts, which may be the result of interactions with industrial fisheries, specifically the consumption of bycatch. Now, the second realm of work is on population community ecology, namely intra and interspecific variation in resource and habitat use. An example is identifying the ecological and physiological causes and consequences of individual diet specialization. Interspecific ecological variation is a really important topic of study because it provides a holistic measure of a species' phenotypic plasticity that integrates ecological, behavioral, and physiological factors to define really what species are capable of. And by extension, interspecific ecological variation is a metric for adaptive capacity that may even have evolutionary consequences and result in speciation. Third is work on ecophysiology, namely what I like to call metabolic plasticity, that is an important component of maintaining protein homeostasis in animals. My talk will focus on this realm to some degree. And it's important to note that ecology informs physiology and vice versa. Physiological capabilities, essentially how animals operate, dictate their ecology, how they interact with their environment. You can't just study one or the other if you want to understand how animals respond to environmental change. So I want to briefly mention some of the big picture questions that guide our research in my lab in ecophysiology. The first is, how do ecological factors like food availability influence the allocation of both exogenous and endogenous resources to animal metabolism? And I'm specifically interested in the growth and maintenance of structural tissues, essentially the things that animals are made of. Second, how flexible are animals in their ability to utilize different types of dietary macromolecules for metabolism and tissue synthesis? So things like protein versus carbohydrates versus lipids. How flexible are they in allocating these different forms of energy into these different compartments? And a much more recent question that we've gotten into is, what role do microbial communities, gut microbial communities, play in contributing to such plasticity? So before I get into the meat of my talk, I want to provide some background on my primary tool, stable isotope analysis, which are stable isotopes are just different forms of the same element that differ in the number of neutrons and thus have slightly different masses. Ecologists often report isotope compositions as delta values, which is just the ratio of the rarer, heavier isotope over the lighter, more abundant isotope relative to some internationally accepted standard. Now, there are several advantages of using isotopes over traditional dietary or even physiological proxies. First, they provide a time-integrated measure of how animals use resources. And the amount of time integration depends on the tissue type analyzed. Some tissues like blood plasma turn over really, really rapidly and record days to weeks of ecological information, while other tissues like bone collagen turn over very slowly and can integrate over weeks to years of an animal's life. Some tissues continuously grow but are metabolically inert, things like whiskers and baleen, so they can be subsampled to provide a longitudinal ecological or physiological record over the scale of months to years. And with any proxy, there are disadvantages, right? Most notably, isotopes do not vary among organisms that occupy the same ecological niche. Rather, isotopes record ecological function. So for example, isotope values likely differ between a benthic grazer in a kelp forest and a filter feeder in that same system. But two grazers likely have the same isotopic composition. But the ability to measure ecological function is still a very powerful approach to study population and community ecology. The last point I want to make with this slide is that isotopes have been used most frequently to study resource and habitat use. But one thing I hope to convince you of today is that isotopes are also very useful for tracing physiological processes, because different tissues, muscle versus blubber, have different isotopic compositions owing to biochemically mediated fractionations that occur in the body. So ecologists that commonly collect and analyze protein-rich bulk tissues, things like liver, blood plasma, muscle, and bone collagen. In our lab, we take this a step further and we break apart these tissues to liberate the individual amino and fatty acids that they are built from. And then doing some fancy wet chemistry, we can prepare them for analysis on the mass spectrometer. We can analyze non-essential amino acids that eukaryotic consumers can assimilate directly from dietary protein, or they can synthesize de novo using a variety of macromolecular substrates like lipids and carbohydrates. And we can also routinely analyze the isotopic composition of essential amino acids that eukaryotic consumers cannot synthesize de novo and must acquire directly from their diet or from their gut microbiome, bacteria and fungi that can synthesize all amino acids from scratch. Last point I want to make with this introduction is that why do we focus on amino acids? Well, for two principal reasons. First is 50% or more than 50% of the carbon and nearly all the nitrogen in the animal is bound up in amino acids, all right? The second reason is that we can lean on a very solid biochemical framework that's been developed over the last 50 to 100 years to help build predictions on how animals assimilate exogenous and endogenous resources and use them to fuel their metabolism, growth, and reproduction. So for the first meaty section of my talk, I will focus on the big picture question of how do ecological factors like food availability and quality influence the allocation of both exogenous and endogenous resources to animal metabolism. And again, I'm interested in the maintenance and the growth of structural tissues that animals are made of. So for this section of the talk, I'll describe the results of some separate but somewhat related studies that are exploring biochemical mechanisms used to maintain protein homeostasis during fasting. Now, marine mammals are an ideal group to work with on this question because they often spatially separate foraging from breeding activities. An intriguing topic, and the physiological mechanisms that allow them to do so have been an intriguing topic of study for many decades because few other mammals are capable of such feats. And fasting plays a really integral role in determining an individual's fitness, namely its survival and reproductive output. Now, while I'm focusing on fasting for today's talk, we are using some of these approaches to examine things like capital and income breeding and the impacts of reproduction and migration on nitrogen balance in organisms. And I'll quickly go over some of those. So let's consider the annual life history of an adult female polar bear. Females with cubs hibernate during the winter. They emerge in the spring to hunt ice seals to replenish their stores, but then they go through a second fasting phase in the midsummer when their primary prey, ice seals, disperses across vast distances of sea ice. As temperatures decrease, hunting opportunities increase just before they descend into the den for the winter hibernation. So polar bears in the 21st century face two big challenges. The first is that the abundance or accessibility even of their preferred prey, like ice seals, will likely decline. And the second is that these periods of fasting, especially the summer fast, will likely increase. So when exogenous resources like ring seals are readily available in the spring and fall, polar bears use these resources to fuel activity and maintain their bodies. Now, the paradigm found in your general physiology textbooks is that dietary fats are primarily burned as a source of energy, while dietary proteins are used to grow and maintain proteinaceous tissues. But when polar bears undergo an extensive summer fast, it must solely rely on endogenous stores in the form of skeletal muscle and adipose tissue for energy, and to maintain protein-rich tissues to remain active in search of food. This is often called a walking hibernation. Now, luckily, proteins and lipids have very different carbon isotope values. Here's some data for polar bears and their prey. Blubber, on average, has carbon isotope values that are 6 to 7 per mil lower than associated muscle. It doesn't depend on what kind of prey species you're talking about. The reason for this is that there's a large carbon isotope fractionation during the conversion of pyruvate to acetyl-CoA downstream of glycolysis, which labels the acetyl group with really light 12 carbon. And these acetyl groups are the backbones of fatty acids. Hence, lipids have really light or low carbon isotope values than co-occurring proteins, even in the same individual. Now, a hint that dietary proteins might be important for maintaining protein-rich consumer tissues was apparent in our work on a wild population of southern sea otters, this one on San Nicolas Island off Southern California. We found a decrease in the difference in carbon isotope values of sea otter whiskers versus the protein in their diet. Okay, whiskers are pure protein versus the protein in their diet as the lipid concentration of their diet increased. Now, this suggests that otters are using dietary lipids to build the keratin in their whiskers. We use the percent of sea urchins consumed by the population, or by each individual sea otter, I should say, as a proxy for dietary lipid content, because weight percent concentration data showed that urchins were by far the most lipid-rich food consumed by this population. So back to polar bears. Then postdoc, now assistant professor John Whiteman, used this idea to examine their fasting physiology with the simple question, do polar bears use adipose lipids to maintain structural tissues during their summer fast? We were primarily interested in the summer fasting period when animals are actively searching for food, but not usually finding a lot of it. So I'm going to show you carbon isotope data for non-essential and essential amino acids of polar bears over a range of body conditions. Okay, more negative values, poorer body condition, more positive values, higher body condition or better body condition. If adipose lipid carbon was contributing to non-essential amino acid synthesis during fasting, then we would expect the carbon isotope values of these amino acids to decrease as body condition declined. That's exactly what we see for these three amino acids, glutamic acid, aspartic acid, and alanine, which combined contribute over 35%, over a third basically, of the amino acids found in skeletal muscle. For the essentials, on the other hand, we wouldn't expect to find a change at all because across body condition, because bears can't make these types of amino acids de novo. And that's exactly what we observed for phenylalanine, isoleucine, and leucine is basically a straight line. And it's important to keep in mind, as I mentioned a little bit earlier, is that the majority of amino acids found in tissues like skeletal muscle are non-essential amino acids, okay? So this physiological flexibility can make a huge difference in the ability to maintain tissue proteins and maintain your body during times of fasting. So just to summarize the polar bear results, they can use onboard lipid stores to maintain protein-rich tissues during the summer fast when they must remain active in search of food. And this has suggested a really inherent amount of physiological plasticity. And the real question is it enough? As this period of summer fasting increases, which is projected to increase with climate change and increased warming of the Arctic, is this kind of plasticity enough to keep them alive as a species? So we're also developing proxies based on amino acid nitrogen isotope analysis to study fasting and reproductive strategies, capital versus income. An example of this is a project with Dr. Nico Lubkern, who is now a postdoc in our lab. We are primarily analyzing whiskers collected from southern elephant seals, adults and pups, who undergo a multi-month fast after they are weaned. Previous work by Nico had estimated whisker growth rates for both pups and adults, so we can confidently tie particular portions of a whisker to an animal's annual life history. So the bulk whisker nitrogen isotope data showed a small but steady increase of about 1 per mil during the post-weaning fast, followed by a precipitous decline in nitrogen isotope values associated with independent foraging when the animals left the beach and were in a anabolic state. So here are the amino acid data. Red symbols represent whisker segments grown during fasting, and blue represents whisker segments grown during independent foraging. The first pattern I want to focus on is that amino acids labeled with a star, nitrogen isotope values were significantly higher during fasting. In some cases, the isotopic differences are huge. We're talking about up to 6 per mil and things like glycine, serine, and proline. This would be equivalent of 2 trophic levels if you believe in the old adage, you are what you eat, plus 3 per mil. As many of you know, mammals have higher nitrogen isotope values than their prey due to nitrogen excretion and isotope discrimination that's associated with the deamination of amino acids and the preferential excretion of isotopically light nitrogen in urea. So for bulk tissues like whiskers, typical isotopic differences between mammal consumers and their prey are about 3 to 4 per mil, like I just mentioned. Now, one reason for this pattern is gluconeogenesis, which is a really critical biochemical pathway for marine mammals, since their diet doesn't contain enough glucose to fuel their metabolic demands. These glucogenic amino acids are deaminated to generate a variety of compounds that can be ultimately converted into glucose. Okay. Some ketogenic amino acids like lysine, I even partake in this process because acetyl-CoA that they're converted to eventually makes it up into oxaloacetate, which can go up to glucose and can be converted into glucose. Amino acids containing light nitrogen are preferentially catabolized for gluconeogenesis, and that light nitrogen is then packaged into urea and excreted via your pee, via their pee, which leaves isotopically enriched amino acids in the bloodstream for the growth and maintenance of tissues like whiskers that we're sampling. So the pools of amino acids used as precursors for gluconeogenesis is switching between one with a low nitrogen isotope value, here is exogenous prey, to one that has a fairly high nitrogen isotope value, endogenous muscle, during fasting. A second interesting pattern is that alanine nitrogen isotope values are actually the exact opposite. They're actually lower during fasting. They drop by about 3 per mil in fasting pups, which is opposite the pattern I was just showing you for the other glucogenic amino acids. We think this has to do with the Cahill cycle, or also known as the glucose alanine cycle, which transports nitrogen from skeletal muscle to the liver using alanine as a carrier, okay, during times of nutritional stress like fasting. Glutamate is deaminated in the muscle. That deaminated light nitrogen is then passed on to transferred to alanine, or transaminated to alanine, which is then transported to the liver via the bloodstream. And this depleted 14N alanine can be used to maintain tissues like whiskers that continue to grow during the fasting period. Now, the last pattern that I want to highlight is the lack of change in nitrogen isotope values of these branch-chain amino acids and glutamic acid, which I think is actually the result of different processes. Branch-chain amino acids are deaminated via a specialized set of enzymes called BCAT enzymes. And the fact that we don't see any changes in branch-chain amino acids might be the form of protein sparing, by which catabolism of these essential amino acids is minimized during fasting, which has been observed in starved mammals in the laboratory. Now, the reason why we don't find a significant change in glutamic acid nitrogen isotope values is probably because this amino acid is one of the most abundant in the body. Most of the nitrogen in your body is bound in glutamate and glutamine. Thus, the overall size of the glutamic acid pool, coupled with the fact that it is involved in a lot of different reactions, it's kind of the center of the nitrogen cycling in your body, helps homogenize its isotopic composition and doesn't result in significant isotopic fractionation between anabolic versus catabolic states in elephant seals. So to summarize the elephant seal results, nearly all glucogenic and ketogenic AAs we measured showed significant increases in nitrogen isotope values with fasting as the animals switched from exogenous to endogenous amino acid pools. Alanine nitrogen isotope values showed the opposite pattern. They decreased with fasting. We think this is a result of the glucose alanine cycle. whereas branch change amino acid and glutamic acid nitrogen isotope values were not sensitive to fasting at all. They didn't change significantly. And we think the decrease in the alanine D15A and coupled with the no change in the branch chain amino acids is why this is a fasting pattern. And when you couple this with increases in the glucogenic amino acids that I showed you first, it provides a new framework for estimating nitrogen balance in wild animals. But it also throws into question the use of amino acid nitrogen isotope values to characterize trophic level and or movement using tissues that are influenced by fasting physiology. So the point here is that beware of using these amino acid tools to look at trophic level or even movement if you're looking only at source amino acids. If you're at all measuring tissues that have been influenced by fasting, and chances are, if you're looking at a marine mammal or even in certain periods, a seabird, there's a good chance during certain periods of its annual life cycle, it's been influenced by fasting, or it's in a fasting state. We're also using this technique. This is a work of a new postdoc in the lab, Dr. Oliver Shipley over here. We're looking at bonnethead sharks, and we're looking at their nitrogen isotope balance across reproduction. These are all females. From early term pregnancy to late term pregnancy, they go through this transition from relying mostly on eggs to relying on placenta. So there's a placental transition that occurs here. We see a significant decline in the nitrogen isotope of the bulk tissues of females across this reproductive, these reproductive phases. And of course, this is backed up by declines in almost all of the amino acids we've looked at, including what I call physiological amino acids, things like glycine and serine, trophic amino acids, glutamic acid and proline, and source amino acids. The only exception here is phenylalanine, which doesn't show a trend. But those are pretty large declines. Interestingly, we don't see any change in what people often use as a proxy for trophic level. So the difference between the trophic and the source amino acids in this early term and late term parts of pregnancy does not change at all. And the preliminary results suggest that that this pattern could be the result of probably two things, and they're not mutually exclusive. The first is that there's been a decrease basically in the trophic discrimination, an example of protein sparing, where sharks decrease their nitrogen excretion rates and you get a decrease in the trophic discrimination between their tissues and their diet. And the other interesting, intriguing possibility that we're actually using as a basis of a grant proposal is looking at nitrogen recycling in these sharks, using remobilizing urea, microbial breakdown of urea, and remobilizing that light nitrogen and shunting it into reproductive effort, which is a pretty exciting opportunity and possibility. We're also looking at this in blue whales. This is another postdoc in the lab, Dr. Geraldine Busquets-Vass. She has an amazing collection of baleen plates from blue whales collected all over the North Pacific, something like 20 plates now, 20 individuals. We see this really cool pattern, of course, associated with migration and things in adult females in this population in the Eastern Pacific. And she can identify different parts of the Eastern Pacific where they're spending time, the Gulf of California, the California Current, or the Costa Rica Dome. You contrast that pattern she sees in an adult female with an adult male, and we call this the lazy males. They just stay within the California Current over several years. They don't migrate with the females. And then what Geraldine's been doing is, again, applying this amino acid approach to look at nitrogen isotope anomalies in different regions. If you look at those anomalies, these are anomalies versus the bulk tissue value, so what I'm showing here. And if you look at a plot of these anomalies, what you'll see is that the females have a lot of anomalies, and mostly positive anomalies. And those positive anomalies are mostly associated with their time on the breeding grounds. So in this particular example, we're talking about an animal that's migrating between the California Current largely and the Gulf of California to breed. And the anomalies that she's seeing in the nitrogen isotope values of individual amino acids are representative or reflect that time in the Gulf of California. So we think this is a reproductive signal. You contrast that with an adult male, there's very few anomalies. They're basically zero all the way across. So we think this is a representative of, or a reflection of reproductive investment. And we're trying to further develop this proxy by using these longitudinal records from baleen. Okay, I'm going to switch gears a little bit here and describes some ongoing work that is focused on quantifying the sources of production that fuel kelp forest food webs. This work blends my interest in population and community ecology with historical ecology. It's developing a tool, essential amino acid carbon isotope fingerprinting, which I'll describe in a bit, for tracing sources of production and consumers that occupy all levels of the food chain. And I'll end this part of the talk with a few slides summarizing how we were applying this tool to study the influence of kelp production over centuries to millennial timescales by utilizing the bones of marine mammals and other top consumers found in coastal archaeological sites. So a recent paper by some folks just up the coast from you, some of them are actually associated, down the coast actually, down the coast from you in Monterey Bay, argued that the presence absence of kelp forest habitat was an important facilitator of sea otter recovery in California for two primary reasons. One is kelp provides cover from white shark predation, which was previously shown to be a major source of mortality for sea otters in this area. And two, kelp provides important nursery habitat, whose absence may limit adult female sea otter dispersal. So our work is asking a slightly different but related question, and that is, how dependent are sea otters on kelp as a source of energy, and how has that changed over the past few millennia along the California coast? This work formed the core of the dissertation of one of my PhD students, Emma Elliott Smith, who recently graduated and now is a postdoc at the Smithsonian Institution with Tori Rick, who's an archaeologist. So is kelp an important source of energy for these consumers? We know kelp serves as an important structural role, providing habitat to many species, like I just mentioned. But how much energy actually flows from these foundation species up the food chain is an intriguing question that is difficult to answer. And of course, isotopes, carbon isotopes, have been used to address this question. The general trend is that phytoplankton, particular organic matter, has lower carbon isotope values than co-occurring macroalgae. And among macroalgae, kelp typically has higher carbon isotope values resulting from decreased fractionation, from CO2 limitation, and the use of 13C enriched bicarbonate as an inorganic substrate for photosynthesis. However, unfortunately, there's a really large degree of overlap in carbon isotope values among these primary producers. One reason for this is that when one measures the carbon isotope composition of these producers, you're measuring a mixture basically of carbohydrates, proteins, and lipids. And the relative amount of carbohydrates, proteins, and lipids in an autotroph like this can change very quickly in response to environmental conditions. So we began to focus on essential amino acids, which I mentioned before cannot be synthesized de novo by eukaryotic consumers and must be directly routed from dietary protein. This routing means that essential amino acids undergo minimal to no isotopic alteration as they are transferred up food chains. Now, a recent paper by Kelp McMahon and colleagues illustrates how little essential amino acids are altered as they pass from prey to consumer. This is a controlled feeding experiment on Gentoo penguins in captivity, fed a single diet item, herring. The data are reported as big delta 13C, which is essentially the offset in carbon isotope value between the amino acids in the penguin and the amino acids in diet. And you can see that the offsets for essential amino acids over here are practically zero, which shows that there is minimal isotopic alteration as they are routed directly from dietary protein into the protein of the consumer, the penguin. So the primary approach we are developing is known as essential amino acid fingerprinting. Now, while differences in the carbon isotope values of individual amino acids are useful, the actual patterns among amino acids might be more insightful, all right? For example, these two producers have different amino acid delta 13C values, but very similar patterns. These are C4 and C3 plants, so things like grass from New Mexico and shrubs like New Mexico. Very different isotope values, raw values, but those lines are roughly parallel, very similar patterns. But these two other synthesizers of essential amino acids, bacteria and fungi, have completely different patterns. And these differences in patterns of carbon isotope values among essential amino acids, that's what we are terming fingerprints. Now, the easiest way to tease these things apart, these fingerprints apart, is with a multivariate approach, like a linear discriminant analysis. Here are data from Larson et al. 2013 showing the carbon isotope fingerprints of essential amino acids of organisms that can synthesize essential amino acids de novo, including fungi, bacteria, terrestrial plants, and various types of phytoplankton and macroalgae. Okay. If you zoom in a little bit on the I should mention these patterns. The main reason why we get these patterns in the isotopes and the fingerprints, the differences in fingerprints, is that these synthesizers are making these essential amino acids in slightly different ways, different biochemical pathways, and those biochemical pathways imprint on their carbon isotope composition. Okay. The second plot here that I just showed you is a blow up of the patterns among marine producers. Now, if you squint, you might be able to see some differences between red algae, brown algae, and phytoplankton. But one thing to note is that these samples were not collected at a single locality, but rather are sourced from a series of random sites in the Pacific and Atlantic Oceans, and some of the phytoplankton are actually grown in the lab as cultures. Okay, so what Emma set out to do is to create a library of essential amino carbon isotope values and fingerprints among macroalgae and phytoplankton at 3 sites in the Eastern Pacific, including Alaska, Gulf of Alaska, Central and Southern California, and Northern Chile. These include green algae, red algae, kelps, and phytoplankton. And note that for kelps, we have samples that were collected in Monterey Bay from 1896 to 2016 by utilizing a historical collection of samples archived at the Smithsonian. And one of our samples was actually collected by Sylvia Earle, which is something we're really proud of. And in total, Emma has analyzed over 150 producer samples spanning 2 continents, 3 major oceanographic provinces that were collected over the past 120 years. This is kind of the take home or the money figure, I would say, from all that work. This is just a linear discriminant analysis, again, a different type, slightly different type of multivariate approach. But nonetheless, you can see ulva, the green algae, kelps, POM, and red algae, different colors. And then the numbers denote different sites, whether they're from Katmai, Santa Cruz, Monterey Bay, San Diego, or Antofagasta, Chile. And you can see that kelp fingerprint separates very nicely from red algae fingerprint. And both of those separate very well from the POM and green algae. Unfortunately, we can't tell the difference between green algae and POM using this fingerprinting approach. But if your question has to do with how important kelp are in a system, looks like a very promising approach to use. That's exactly what Emma did. This is an example of one of her chapters from her dissertation. This is work done down in Antofagasta, Chile with colleague Chris Herod. Interestingly enough, in this particular system, kelps and phytoplankton essentially have the same bulk tissue values. Okay, so this is an example where there's a high degree of overlap in the bulk tissue values between these 2 major sources of energy in this system. But when you do the fingerprints on these same samples, which is what Emma did, she showed that you can separate out kelps from red algae. Again, a little less discrimination between green and POM, but essentially kelps are different than all the other sources. Okay, and you can basically tell them apart. Emma took this a step further, and she looked at the importance of these 2 different sources or 3 different sources of energy or energy channels in individual consumers, and found some evidence of individual specialization in energy channels for some consumers. So it's a really cool example of how organisms are specializing on particular diet items, but in essence, what they're doing is they're specializing on different energy channels, whether it be this fast phytoplankton energy channel or this more slow kelp macroalgal channel. Now, we're also extending this approach back through time, which is Emma's true passion, by analyzing the remains of invertebrates found in archaeological sites. And present, we're focusing on the remains of sea otters and sheephead, 2 keystone species that fill the same ecological niche in Southern California kelp forests. And we have some preliminary data from archaeological sites on San Miguel and San Nicolas Island that span the last 3,000 years. Now, if we assume that essential amino acids are not significantly altered as they are transferred up the food chain, we can directly plot the fingerprints of modern and ancient consumers on our producer fingerprints from producers from this site. And this is our LDA. Again, I'll just remind you, it looks slightly different than the ones I've been showing you because it has a different group of samples in it. This is local samples. Kelps and red algae separate out very nicely, but again, POM and ulva have a high degree of overlap. Now, when we plot ancient San Miguel sea otters, so these are sea otters actually taken from the picture of the site I just showed you. This is a site that's about 500 years old on the west coast of, or the west side of San Miguel Island. They show a very strong kelp signature. About 76% of the samples we've analyzed so far classify with kelp. Same with sheephead. So these are sheephead bones taken from the same archaeological site, show a very strong kelp signature. 12 to 14, or about 86% of them, also classify with kelp. And ancient San Nic sea otters. So these are sea otters collected from an archaeological site on the west coast of San Nicolas Island, also show an overwhelming kelp signature. Everyone that we've analyzed thus far has classified with kelp. If you compare that, unfortunately, we don't have data for modern sea otters from these islands because they don't really occur there in very high numbers. We do have data from the adjacent California mainland. The sea otter population is beginning to, they've passed point conception and they're beginning to work their way down the Santa Barbara coast. And we'll have those data pretty soon. But we do have modern sheephead, and this is actually courtesy of Scott Hamilton's work and who we're collaborating with on this. And they're a lot less kelpy than their ancient counterparts. Only 43% of the modern sheephead that we've analyzed thus far classify with kelp. There seems to be a lot more variation in their energy channels. Now, we can use a mixing model approach to quantify the importance of kelp for these ancient consumers, for both ancient otters and sheephead from San Miguel and San Nicolas Island. And if we do that, about 80%, depending on where you are, of their essential amino acids were ultimately derived from kelp production. So the kelp summary here is that major sources of micro and macroalgal production have very different essential amino acid fingerprints that are consistent across space and time. And the reason for that is that they're driven by conserved biochemical pathways. I mean, these patterns are a reflection of differences in biochemistry between these different producers. And so we wouldn't necessarily expect that they would be, they would change spatially or temporally. And that's kind of what we've seen with our modern library. These patterns provide a way of quantifying with high resolution the importance of kelp as a source of energy from primary to tertiary consumers. If you assume again that essential amino acid fingerprints aren't altered all that much as you work your way up a food chain. And in a marine food chain where, you know, most things beyond the first step, most things have a very high protein diet. Even the first step, for that matter, a fairly high nutritional quality of algae in comparison to more terrestrial systems that have a lot of recalcitrant compounds like cellulose, where gut microbes might play more of a role. I think this is a pretty good assumption in that these essential amino acid fingerprints are passed up the food chain. Preliminary data show that kelp was an important source of energy for ancient Channel Island consumers. Ancient sheephead are more kelpy than their modern counterparts. The otter data is coming soon from the adjacent California mainland. Emma still has a little bit more work to do. This is actually for Emma 5th chapter of her dissertation that she's still working on well past graduation, which is fine. I'm never gonna give student a hard time for working on their 5th chapter after graduation, but we hope to have that data wrapped up this year and submitted. So this data suggests overall that this approach, essential amino acid fingerprinting, could be very useful for studying energy flow and energy channeling in very complex aquatic food webs, including coral reefs, estuaries, rivers, and lakes. And I just want to give you 2 or 3 examples of other systems we're working on where we're applying this approach. The first is work done by a PhD student in our lab, Alexi Besser, who's interested, she's an aquatic ecologist, is interested in autochthonous and allochthonous resources in rivers. So autochthonous resources being largely algae and diatoms that grow in the river, and then allochthonous resources being the terrestrial organic material that sort of gets washed in the river every once in a while when the river floods, okay? So she's created another library, this one of a terrestrial library and freshwater aquatic library, showing very nice separation in fingerprints of things like C3 plants, C4 plants, and algae that she can apply here. And Alexi wants to take this a step further because she's also interested in the role of microbes in this system. And this is an old idea. It's called the peanut butter and cracker hypothesis. It goes back to the 1970s when it was first mentioned in a paper. But the crackers are terrestrial plants, okay? Very recalcitrant cellulose rich, sometimes even lignin rich material. And the biofilm is the peanut butter. And the idea is that the biofilm needs to break down and convert that recalcitrant terrestrial organic material into compounds that are much more readily accessible by aquatic consumers, things like aquatic invertebrates, like, midges, chironomids, that kind of stuff. So Alexi's trying to figure out if there's a microbial, sort of a biofilm loop here that synthesizes amino acids de novo from this recalcitrant terrestrial material that is then fed up the food chain in these river food webs. And the last example I'll mention is work actually done by your very own Mike Fox, who was a master's student with Mike Graham before going down to Scripps and getting his PhD. Mike's now a postdoc at Woods Hole, studying corals. This is work that Mike did with Emma and myself and his PhD advisor at Scripps, Jen, on corals during his dissertation. And Mike was interested in the relative influence of autotrophy versus heterotrophy in corals. And we came up with this idea of using this fingerprinting approach to tell essentially essential amino acids that were synthesized by the zooxanthellae versus that the corals are hosting, versus essential amino acids that are synthesized by phytoplankton that form the base of the heterotrophic nutrition that corals might rely on. And we found really nice separation in those two sources of essential amino acids for the coral animal. And then we analyzed a bunch of coral animals. This is actually at Palmyra Atoll in the South Pacific. And we found that there's a variety of different dietary strategies from near 100% heterotrophy, like over here, all the way down to very little heterotrophy over here. And this is really interesting because these individual coral animals were collected on the same reef, sometimes meters apart from one another. So even within the same small area, same population, of course, they have a individual animals have very different strategies for making a go of it. And this also has implications for coral bleaching, because what happens during bleaching events is that corals sort of discard their zooxanthellae. That's why they turn white. But they do have lipid stores and other types of stores that they can survive bleaching events for fairly long periods of time, as long as those stores are long enough or get them through. And so the idea is that if we could come up with a proxy for heterotrophic nutrition in corals and how much they rely on heterotrophic nutrition, that might be a good indicator of whether or not a particular reef or a particular species has a chance of rebounding from big bleaching events. And also there's a spatial component as well, because different parts of the reef have, based on their proximity to the ocean, have different availability of heterotrophic nutrition as well. So I'll stop there. I just want to thank my collaborators, John Whiteman, a bunch of postdocs and students and colleagues from around the world that have aided in this research, a variety of different funding agencies where we get our funding and our money, and I'll take any questions. Thanks.
Okay. Well, thanks. That was that was an impressive breadth of different types of studies. The compound-specific isotope stuff has always been intriguing to me, and I didn't quite understand, I guess, the fingerprint side of it, but I have to say that how you presented it certainly made a lot more sense to me. The one argument or the one example that you showed with the corals, I thought was really pretty interesting with the heterotrophy and the use of the zooxanthellae. And I was thinking the exact same thing when I saw that you could imagine some of these corals might be sort of shaded, if you will, on the reefs by other corals or other structures, and so some might have to rely on food particles and things coming to them versus the sunlight. But it's interesting that you also countered that by saying that they were from the same almost the same spot, so it's really quite a unique situation. I guess it was more of a comment than a question, but
yeah, I mean, I think there's a lot to explore there. I mean, we were amazed to see that pattern. We actually went into this thinking that the zooxanthellae and the phytoplankton the autotrophic and heterotrophic sources would not have different fingerprints, but we were pretty surprised at the result. And then the second result that was really surprising was how much variation you see in such a small area. And the next step for that kind of work, although it's been kind of sidelined because of COVID, but is trying to go out to a particular reef or a set of islands. Actually, Mike had a big idea of going across islands in the South Pacific that have, based on satellite data and chlorophyll concentration data, have different accessibility or different amounts of heterotrophic nutrition available. And I think that's the next step, to see how, over what spatial scale you get this kind of variation. And in certain areas, do you have mostly heterotrophs, and in other areas you mostly have autotrophs because of their access to. And then of course, what are the implications of that when you have bleaching events?
Yeah, I was also thinking too that if you were looking at corals, so in the inner lagoon versus on the outer parts of the reef, you might see differences based on, you know, what can come into them and so forth too. But I don't know if that factor into part of this that you're talking about.
We didn't get, that was one thing we were interested in doing. We did have, I think, 4 or 5 sites around Palmyra, and we saw that there was no real significant difference. But we didn't have an in lagoon site at the central lagoon there. Most of our sites are out on the outside because the species of coral we were focusing on doesn't really occur much in the lagoon per se. So yeah, it was mostly focused on the periphery of the island. And our idea was to go then, you know, to scale it up basically from there to look at across various islands. There's a huge gradient in productivity, I'm sure you know, Scott, in that part of the Pacific as you go away from the equator. And it's an upwelling regime.
Yeah, there's a mass island effect around Palmyra that, yeah.
And so that we wanted to look across that north-south productivity gradient, but it's a proposal for a post-COVID world.
Yeah.
No one's going to Palmyra anytime soon.
Oh, well, I'm actually supposed to be going hopefully in August, but we'll see.
Okay. Well, I'll check back and see if that's happening, because then we might submit our proposal. Yeah.
So questions. Anybody else have any questions? I see Scott Hamilton has a question, and at the request of the speaker, he asked to see faces. So
I want to see Scott.
Perfect.
Scott.
Here I am. What's up, Seth? Good to see you.
Good to see you. How are you doing?
I'm doing great. Yeah. First, I just want to say, I mean, thanks for coming and speaking. You know, you have such a mastery of stable isotopes. I mean, you know, I dabble occasionally, and it's such a useful tool, and I learn something new every time you talk about it. So I really appreciated the seminar today. And I also appreciate all the help you've given to, you know, multiple Moss Landing students, you know, recently that are interested in using these tools. So it was great to see the sheephead data. Yeah, so of course, you know, I'd be excited to see the modern and the historical comparison and how their role might be shifting.
Emma's actually back. I would just give you an update on that. Emma's back in the lab now. She was out for a while. She moved to DC, and then she got sent back to New Mexico because of COVID. Basically, the Smithsonian shut down just a few weeks after she got to DC. And so she was out of the lab. We couldn't have visitors because technically she wasn't a student anymore. So now she's back in the lab, and that's her main goal, is to get that. So you'll be hearing more about that. You'll be hearing more.
Yeah, well, I was just thinking, because we have, I mean, also from those modern samples, you know, we have a lot of information on their size, right, and where we caught them, and their diet from the gut content. So it'd be great to see the Kelpie versus the, you know, the non-Kelpie ones, and what other factors maybe kind of might line up there, right, with all the isotope work to kind of explain those patterns. So that's really cool. My question was actually going to be about the polar bear stuff. Right, so there's evidence that this summer fast is increasing, right, with climate change. But my question is about the winter hibernation period. Is there any evidence that is getting shorter, I guess, so that they're having more time maybe during that early spring foraging to kind of catch up? Or
I think I don't know the specifics of that. I think most of the work in terms of physiology is focused on the summer fasting, because they're worried that period is going to increase. And with sea ice loss, the accessibility to prey is going to be much reduced. And so there's a lot of work done on the summer period of, like, can they be basically can they incorporate more terrestrial resources into their diet to make a go of it? Because a ice seal abundance or accessibility is going to decline. Can they provide, or can they rely on marine mammal carcasses that are actually killed by, that are harvested by Inuit hunters? Those kinds of questions. I don't know of anybody that's working on the expected decrease in the hibernation and how that's going to impact reproduction and, you know, cubs and things like that. But I think that's an interesting question for sure. Yeah, but that's definitely one of the time periods right as they come out of the den that they need resources. And so if they're not available, that's a big issue. That's a huge issue, a really critical part of their annual life history.
Well, it seems like isotopes are another tool to answer those sort of questions too, right? How they're shifting their diet, and then potentially what's happening right when they.
Yeah, I know isotopes have been thrown at the question of whether or not they're using terrestrial resources, because that's like one of the bread and butter applications of isotopes. And it's also been thrown at the idea of them consuming carcasses from harvested marine mammals, mostly bowhead whales that are hunted along the Beaufort Sea and up far north. So that's something there, but I don't know about that earlier period.
Yeah, cool. Thanks. So good to see you.
Yeah, it's good to see you too. You have 2 kids now?
Yep.
Okay. Same.
Keep me busy.
Yeah. Last time I saw you in person, it's very different world.
Yeah, life has changed. We need to go to Chile again and teach that class.
Yeah.
Yeah.
Okay, so let's see. We have another question by Matt Hess. So if you want to share your video, that'd be great. All right.
Hi, Dr. Newsome. My name is Matthew Hess. I'm in the Ecology lab with Mike Graham. And I've actually been pointed your direction a couple of times because I'm really interested in stable isotope analysis, both by Mike and Mike. Mike Graham and Mike Fox have both told me to get in touch with you. So nice to meet you.
Nice to meet you.
So I'm particularly interested in nitrogen supply in giant kelps and looking at stable isotope, isotope fractionation as a potential way of determining sources of nitrogen in kelp, and was particularly fascinated by your work where you're able to isolate fractionation signals among different autotrophs. I thought that was really cool. And I'm wondering if you're familiar with anything that's happening with stable isotopes within kelps, maybe across a population, or just looking at metapopulations across a geographic or temporal scale or anything like that.
I don't know. I mean, Mike did a really nice paper with isotopes during his master's, looking at carbon isotopes and the effects of growth on carbon isotope fractionation. Mike Fox, I should say. I don't know of anything that's similar to that with nitrogen. And I think it's a kind of an open question. I think there's a lot, there's a lot of work that's been done, and you should look at papers by Falkowski. There's a few folks that work on algae and isotopes in algae specifically, but most of that work, to my knowledge, has been focused on carbon isotopes and photosynthesis and growth rate and the impacts of those things, the interaction of those things. I think nitrogen would be fascinating to look at. I think one thing, off the top of my head that Mike and I have had conversations about in the past are like, kelps have this ability to store nutrients for long periods of time, and how they reallocate those nutrients for growth is kind of an open question. And we had some ideas about carbon isotopes and how you could use that with the essential amino acids with Emma's work, because Mike's involved in that. He's actually a co-author on that library work. But we never thought about nitrogen. But there's definitely some possibilities, and I can think of some things. One of the things that people look at is like the offset between different types of amino acids. They call this a beta value. So you look at an essential amino acid and a non-essential amino acid, and there's an offset there. And that might be reflective of different growth conditions or different sources of nitrogen available to the plant, or I keep saying plants, but you know what I mean, to the algae. So that, I think it's an open thing.
Very cool. Very cool. Thank you.
Okay, so it looks like Mike Graham has a question. Mike, do you want to unmute yourself?
He's thinking about it. Oh, there he is.
There we go.
Hi,
Am
Mike.
I on there? Can you hear me, Seth?
Yes. How you doing?
Great, buddy. Great to see you.
Yeah, it's good seeing you.
Man, it feels like a reunion. Good question, Matt Hess. And I got something. You know, you brought up the ghost of Mike Fox. I mean, it's not been that long, but you know, his time here at Moss Landing was unique in that contribution. And if you remember in that paper, he was showing different parts of the kelp plant had different carbon signatures, right? Which was really the essence of that. And because of that, it's really hard to just use straight up carbon isotopes to look at things like particulates, especially going into the detrital part of the food web, because you can be putting particulates out on a carbon, you know, on a heavy blade or on a lighter blade. And so it really makes your you know, signature story you set us up with, where it was kind of muddied by the overlap, it makes it even worse. Is there a potential for your essential amino acid specific work to help with that? Or do you see the same problems going to show up if you look at particulates of kelp? Even though the kelp fingerprint might be tighter, what happens when they make particulates? You know, I mean,
yeah.
Does the fingerprint differ from a growing part of the kelp versus a non-growing part of a kelp?
Yeah. So I know the off the cuff answer to your question, I think it's a good one, is that I don't expect. So we looked at that actually with some, I think we had collected them down in La Jolla. If I remember correctly, Mike had collected them during his dissertation, but we looked at different depths essentially. And we tried to look at fingerprints across the depth to see if we really wanted to see if this fingerprint was true, regardless of growth conditions, regardless of light conditions, even nutrient availability. And we found that actually we have a really cool picture of Emma with the whole plant that we took out and put on Scripps Pier and sampled. We found no changes in their fingerprints across depth.
Wow.
And but the open question, I was part of a thesis PhD qualifying exam at Santa Cruz 2 weeks ago with Mark Carr and Pete Ramundi and a student of theirs, Casey, and the question came about because we study brown and green food webs and terrestrial systems, and the question I had for Casey was this particulate matter, this detrital source of food in these systems, how much of a microbial influence is there in the breakdown of that food? Because I think a lot of marine ecologists think of detritus as just flotsam and jetsam pieces of kelp with various particulates or whatever sizes floating around on the bottom of the ocean. And I was wondering about the microbial influence and how much microbes break down that material. And I would say if there's a huge influence of microbes in the breakdown of that material, then you certainly could change its fingerprint, because what you're doing is you're converting the algal biomass into microbial biomass. But if it's just straight shredding, you know, of blades into smaller and smaller and smaller bits with no microbial assistance, then you're probably not going to have a big difference in the fingerprint between you know, a live blade and something that's been shredded up.
Well, that's really cool, because underneath all of those bones in your middens that you've got on the islands are a bunch of abalone that are eating both detrital and non-detrital kelp. But more importantly, you got a bunch of middleists, right, that are great filter feeding little indicators of what's been going on. And as you remember, there's some hypotheses that kelp was more or less abundant over millennial timescales out there. So that would be a really cool way to look at those scales you're talking about for Palmyra and the role of heterotrophy versus, you know, in the kelp forest, the role of the detrital pathway, you know, the direct detrital, because I don't think you're going to get a lot of breakdown that fast. You know, they slough off the particles is the cool part. You're not talking huge pieces of kelp that get broken down to smaller and smaller pieces. They're already sloughing off edible pieces that are probably entering that detrital food web fast. So that would be really cool, you know, with the kind of stuff you guys are doing with Tori and all, to really bring that together. And it sounds like there's a lot of promise. And I'm really glad, because as Matt Hess pointed out, I'm constantly bringing you up, saying that you've got the ticket, man. You've got the future for doing this, and it's not as messy as it used to be. So I'm really stoked that I'm not just blowing smoke for everybody.
No, no.
And that you're out there doing it.
Yeah, well, I mean, for me, it's a wonderful. I always, I love the ocean, and I live in New Mexico. So, my main, I mean, I grew up next to the ocean in San Diego, and my main sort of pathway to get to staying involved in marine work has been through visiting students and postdocs and colleagues. So I'm always encouraged. I'm always excited about getting involved with marine-based projects. So that's just a note to all the students out there. Like, if you have a question about isotopes, let me know, because I constantly like to think about, or how they can be used in marine systems. Get in touch with me, and I like to think about that. And I just don't get enough of that in my newly adopted land in the desert.
And thank you for not asking me how many children I have. Take it easy, friend.
Yeah, it's good seeing you, Mike. Good seeing you.
Okay, so while we still have some time for any last minute questions that people might want to ask. I guess one thing I'll ask you about is, I know that you have to build up these libraries of samples to sort of draw your comparisons to what you're collecting from the animals and so forth. And so how much of that is taken up of your time and money versus what you actually, because I'm sure it can be quite large in terms of your catalog and everything.
Yeah. I mean, I think at this point with this technique, like the fingerprinting technique is a good example. I think, and even like using amino acids and nitrogen isotopes and amino acids to look at trophic level, when you're at the like kind of the beginning of the curve as far as their use and their application, I think it's really, really important to do to make those kinds of libraries, because one of the essential questions we have is like, how ubiquitous are these patterns over space and time? And, you know, if we want to apply them back to 100 years ago or 1,000 years ago, you know, can we make a library where we're involving, you know, analysis of museum collections and looking over large spatial gradients? Because I think it's really important to do that. I think maybe 5 or 10 years from now, it might not be. There might be enough of a library out there we could just lean on these patterns. And I'm starting to get more and more confident the more data that we generate on these libraries, that these patterns are, you know, fundamental biochemically driven patterns that we wouldn't necessarily expect space or time to mess with. But I think it's an open question. And I think at this point in the proxy development, I think you have to do it well.
Right.
And it's cool. Okay. What the cool thing about it, though, to be honest with you, and this is more kind of a comment for students, is that it exposes you to biochemistry and it exposes you to physiology, and you have to think about, you know, what creates these patterns at the producer level. And so let's say you're interested in sea otter ecology, or you're interested in blue whales, you need to like think about, that's what isotopes kind of do, is they force you to think about physiology and biochemistry in different organisms. And I think that's part of the fun for me, to be honest.
Right. Okay, it looks like we have a question from Anne.
Yeah, hi. I guess maybe this question is a little late, because I'm a student in the Ecology lab, but I've done a lot of work with museum collections and herbaria, and looking at the work you mentioned about sea otters and their kelp relationships and sheephead and their kelp relationships, I was wondering, in addition to archaeological digs and research, if you had sampled any museum specimens or museum herbaria of kelp to look at how stable like the kelp or other algae patterns were through time using herbaria samples in addition to fresh kelp and other algae samples.
Yeah, that's a great question. And Emma did a little bit of that for her dissertation. Those specimens from the Smithsonian, those were all Macrocystis. Unfortunately, we only had specimens of Macrocystis because it's sort of a charismatic macroalgae, if you want to call it that. It's a cool organism for sure. So you did a little bit of that. I don't know of any other examples, Anne, of people doing it, but it's a huge resource out there. I know Smithsonian's collections of macroalgae, a lot of which were actually collected in coastal California, many at Hopkins Marine Lab, for example, or in Scripps, or up in Friday Harbor. These collections are useful, and we at Smithsonian we've had very little problem. Tori Rick, who's a archaeologist there, has helped to facilitate this, but getting access to those collections and sampling them for isotope analysis. And with a dried sample like that, as long as it's preserved correctly, you don't have to worry necessarily about microbial degradation and kind of any contamination. And so that's what Emma did, and I think, to my knowledge, that's like the only data that I know that exists for, at least at the compound specific level, for macroalgae. But I mean, I think it's a big opportunity. Like, I think you should consider it further. I just never, I've done museum work a lot with animals, and I never, until Emma came along, considered, oh geez, there's also these herbarium collections that can go back decades or centuries in some cases, that can be used as a resource. And when you're doing historical ecology or paleoecology, those kinds of collections are really useful because you're not going to find them in archaeological sites. You don't find pieces of kelp in archaeological sites. You don't find pieces of plants generally in paleontological sites. But if you can find them in museum collections, it gives you a nice resource. So I definitely encourage you to do it. It's pretty easy to do. I'm sure California Academy has an amazing collection.
Awesome, thank you.
Okay.
Oops, Scott, you're muted.
Okay, now I'm good. Okay, so we can open it up now if people would like to stick around for a little bit and talk with Seth.